Okay, so welcome to this second video on excitation-contraction coupling in cardiac muscle cells. So we've actually seen the process of excitation-contraction coupling already. This is the process of excitation-contraction coupling, the coupling of the excitation of the membrane, the changing of the electrical potential differences across the membrane, to the release of calcium, which is then from the intracellular stores, which is then going to cause contraction. And again, I want to stress that this calcium coming through from the extracellular fluid, that the role of that is to stimulate the ryanodine receptor 2 and trigger release of calcium from the intracellular stores. It is not, this calcium does not, it's not responsible for the contraction of the muscle. Too little calcium comes in here to actually cause contraction of the muscle. It's the calcium released from the intracellular stores which drives contraction of the muscle. Okay, so in this video what we're going to do is we're going to look a bit more in detail at this type 2 ryanodine receptor. Okay, so just like the type 1 ryanodine receptor, it is a giant. So I will draw it as a giant great receptor. And again, just like the type 1 ryanodine receptor, it is a tetramer basically, like so. So it's made up of four subunits basically, which are all separate polypeptides. So these 4 portions are separate polypeptides. So you have 4 polypeptides coming together to make this ryanodine receptor 2, and it's often denoted RY for ryanodine, R for receptor, and then 2 for denoting it's the type 2. Now, each one of these subunits that makes up this ryanodine receptor type 2 is identical. So it's a homotetramer, and all four of these subunits are encoded for by the same gene basically, and that's the ryanodine receptor 2 gene. So there is one gene coding for the subunits which make up the ryanodine receptor 2, and you put all four of the identical gene products of that gene together to make a ryanodine receptor 2. Okay, so again, these subunits are absolutely massive. Each one has around 5,000 amino acid residues. So that's a pretty massive protein. And the next thing to discuss is the selectivity pore of these channels. So if I draw like a slice of this, so effectively what I'm doing is I'm imagining taking a slice in this plane here. So I'm imagining putting a plane there, slicing through this channel, and having a look at what it looks like through. And basically what it looks like is it has a wide portion cytosolically, and then it has a thin portion on the lumen of the SR side. Okay, so this is the selectivity filter of our ryanodine receptor. Okay, but compared to the selectivity filters of other ion channels, this is very, very wide, and in fact it's not awfully selective, but just like in the case of the ryanodine receptor type 1, that's actually going to work to our advantage, as we'll see. Right, so this is the selectivity filter of the ryanodine receptor type 2. Selectivity filter. That is supposed to ensure that only calcium moves through it, but we'll see that it's far too wide to actually do that, so I'll draw a tiny little calcium ion going through it. So this selectivity filter isn't actually a very good selectivity filter. But that works to our advantage because it means that the conductance of calcium out of this channel, when this channel is actually open, is extremely rapid. And because we want contraction of the muscle to be extremely rapid, that's a good thing. The other thing to say is that this selectivity filter is not actually completely selective to calcium. It also allows potassium ions to go through, so I'll draw this potassium ion in blue here to distinguish it from the calcium ion. And basically this is very, very important because let me just label this diagram up actually. This is the lumen of the SR, this side. So the selectivity filter faces onto the lumen of the SR. So it faces this side here basically. It's closer to the lumen of the SR, and this is the cytoplasmic side here. So this is the cytoplasm. Right, okay. So basically what happens is as you release calcium from the sarcoplasmic reticulum, calcium is going to leave the the sarcoplasmic reticulum. It's going to go into the cytoplasm, and it's going to be driven by the concentration gradient, because calcium is very high within the endoplasmic reticulum and low in the cytoplasm. Okay, remember there's no electrical potential difference across the endoplasmic reticulum membrane, so there's no electrical driving force across that membrane. Okay, so calcium is going to have a net movement out. The problem is that if you're just moving calcium ions out, you're moving positive charge from the lumen of the sarcoplasmic reticulum into the cytoplasm. So you're going to overall make a negative charge, you're going to leave a negative charge in the sarcoplasmic reticulum, so after all that, negative, and you're going to make a positive charge in the cytoplasm. So you're very quickly going to reduce the electrical potential within the sarcoplasmic reticulum and increase the electrical potential within the cytoplasm. So you're going to create an electrical potential difference across this membrane. Calcium is a positively charged ion. It's divalent. It's doubly positively charged. It's going to want to be where the electrical potential is lower, i.e. more negative. So it's going to want to remain in the sarcoplasmic reticulum if you build up this electrical potential difference, which will eventually get so large that it stops all movement of calcium through this channel. Now, that will be detrimental to us actually achieving contraction of this muscle. So, what you need to do is have some sort of movement of charge to balance the movement of positive charge out, and that's the movement of potassium in. So basically what happens is this channel also conducts potassium into the sarcoplasmic reticulum, as so. So the potassium ions can also go in the opposite direction into the sarcoplasmic reticulum, basically. And potassium ions have a positive charge, so this is positively charged. So this is effectively going to act as the counter ion for the movement of potassium. You are moving, for the movement of calcium. You are moving calcium out of the sarcoplasmic reticulum and into the cytoplasm. So you now need to move positive charge back into the cytoplasm, back into the sarcoplasmic reticulum to replace the positive charge you removed. And that positive charge is going to be in the form of potassium ions, basically. And that's why it's important, actually, that this ryanodine receptor 2 isn't actually selectively permeable just to calcium, and in fact it will allow potassium to move in the opposite direction. So, the ryanodine receptor 2 allows calcium to leave the sarcoplasmic reticulum, potassium to act as the counter ion and move back into the sarcoplasmic reticulum, and then you'll release the calcium into the cytoplasm, and that will then lead to the contraction of the cardiomyocyte.